Morning, Aaron. How are you? You're muted.
Sorry, how are you doing?
Good, I'm good. How are you?
Good to see you.
So I'll be the first one to talk, right?
Yes, please. Do you want to test out your slides, please?
Okay. So let me try to show some video I want to test whether it can play. Can you see my slide?
Yep. Looks great.
Great. Just test one of the videos later. Yeah, I use your reviews cartoon to demonstrate.
Oh, cool. Nice.
Let's see. So the video works as well. Great.
Yep, looks good.
Okay, let's just stop sharing. So far, you enjoying your sabbatic in China?
Yeah, it's great.
So you will leave next month back to Europe?
Yes.
Yeah. To the whole family or just yourself?
That's a good question, but for now, right now the plan is just myself.
Okay,
but we'll
only
see
for you.
We'll see how things go in the next week or so in Shanghai. So is Singapore completely open now?
Kind of back to normal, yeah. The mask indoor is still it's required, but outdoor is optional. And other than that, I think everything is open.
Mm-hmm. I was thinking of, when the lockdown started, I was thinking of coming to Singapore just because it's the same time zone, but with the kids, but it didn't work out.
Yeah, you should visit us again. Yeah.
Always I want to. Thanks. Before COVID, I used to come a couple times per year. I love going to Singapore.
Yeah, I remember you gave a talk at Duke and also to Mexico.
Yeah.
Karen.
Hey, it's Long.
Hey, Mian. Nice to see you.
Yeah.
Hey.
Nice to meet you.
Hi, Hong.
Hey.
Yeah, you're a lot of Singapore fans here.
Yes, sorry. I was trying to unmute but then turn off the video.
It's okay. Yeah, it's okay. We're still waiting for Lee, the other speaker.
Okay. Would you be able to go out for a few hours around?
I have a pass. Yeah, I got this.
Oh yeah.
I got this pass. Yeah.
Yeah. Okay. It's different from us, but it's similar.
Yeah. I went last night and the guard stopped me. I was with my dog. I was like, "Can I go out?" And he's like, "No." I said, "Well, my dog wants to go out." He's like, just walk around your neighborhood." I said, "Well, she was saying she wants to go see other things." Like, no, just come back tomorrow. But this one, they left it blank. So last one, they told us the time. So this one, they sort of left it blank. So I think that's a good sign.
Yeah, that's great. That's great. Yeah, we don't have that yet. We have very limited time. So.
Yeah.
Hi, Jean. Oh, Lee's here.
Hey, Lee.
We're not connected.
Oh. When do you get to go out, so?
We were supposed to yesterday morning, a few hours ago. That's first time.
Yeah.
Hoping that's
But then they told us like when you get back, you have to take an antigen test to get back in. But I was trying to explain it doesn't work exactly like that, but they just need you to do it.
Yeah. For biologists, it's ridiculous. Anyway, you have some kinds of you would have everything would happen in a few hours. So
yeah.
Wait, meaning you can go out on the street or go out of your compound living?
You
can go out of your compound.
On the street. Yeah. On the street for a few hours, then you can take an antigen test. For biologists, it's weird, right? You wouldn't be able to virus quickly infect you, but anyway, it's funny. But how to explain, right? I feel difficult to explain to them. Doesn't mean anything.
Yeah. Lee, do you want to test your slides?
Sure.
Yeah, you can just share them.
Okay, great.
Looks good. Looks good. Okay, and then Liao, if you could stop sharing, I have some other slides, and then Mian will give his talk first, and then I'll introduce you. Thanks. All right, Zalong, should we get started?
Sure, go ahead. Yeah.
Okay. Hi, everyone. Welcome to another Neuro Zoom, or Shanghai Lockdown Edition. I hope everyone's doing well. And before we get started with the talks today, we have more great talks coming next week. We have Zhu Huang from Jiao Tong University and Wei Lu from NIH. So please tune in and check out the webpage. We have great talks this spring and into the summer. We have slots available. So if you have a great story, want to talk about it, let me or Zalong know. You can check out the YouTube channel. You subscribe if you want to watch the talks on YouTube or tune in live. And with that, Zalong will introduce our first speaker, Mian.
Great. It's my great pleasure to introduce Professor Mian Cao, who is now in Duke-NUS in Singapore. Singapore University, National University of Singapore, have joined the college with Duke. So Mian has got his bachelor degree from Xiamen University. After the college, he went to Hong Kong to do neuroscience PhD with the Xiajun and the University of Hong Kong, HKUST, University, Hong Kong University of Science and Technology. Also, June will soon be joining the Neuro Zoom in a couple months, maybe July. June will also talk about his work. So after neuroscience PhD, Mian went to US, did a PhD in Yale with in the presynaptic big shots, the petrol decameron, where Mian did a very unique angle to study the Parkinson disorder, working on a series of presynaptic molecules. So in 2018, Mian come to Singapore to start his own lab in Duke-NUS. Today, Mian will talk about his latest about dysfunction of synaptic endocytal trafficking in Parkinson's diseases. Okay, welcome, Mian. Take it away.
Great. Sharing my slide. Can everyone see my slides well and hear me?
Yeah, I believe so. It can full screen now.
Great. Yeah. So it's full screen now.
Just a second. I believe it's coming to
Looks good.
transforming. Yeah. Okay. Good. Yeah. Sorry. See it. Go ahead.
Great. Yeah. Hello, everyone. And I'm Mian from Duke-NUS Medical School in Singapore. First, I'd like to thank Zilong for the nice introduction, and Zilong and Aaron for organizing this wonderful online seminar series. I've learned and enjoyed a lot of the previous talks, and also very happy to have this opportunity to share our research today. So as Zilong mentioned, my lab is interested in the cellular mechanism of the synaptic endocytic trafficking in neurodegeneration, particularly Parkinson's disease. So I will start with some brief introduction about PD. As you all know, the PD is the second most common neurodegenerative disorder, mainly affecting the movement function. So the motor symptoms is mainly due to the very selective loss of the dopamine neuron in the midbrain region called substantia nigra, and also the dopamine deficiency in its target region, striatum. Another pathological hallmark of PD is the formation of the Lewy bodies and Lewy neurites in the surviving dopamine neurons, which contains alpha-synuclein aggregation. So the genetic studies have identified over 20 PARK genes that is linked to the familiar PD. For example, alpha-synuclein and LRRK2 are linked to the dominant forms, and the Parkin, PINK1, and the DJ1 are linked to the recessive forms. In addition, the GWAS study have also identified so far over 90 novel risk loci in the sporadic PD, as shown in this table. You can see the alpha-synuclein and the LRRK2 actually are also the top hits, suggesting both familiar and sporadic PD, they may share a very similar cellular and pathogenic mechanism. So there are several cellular pathways involved in the PD pathogenesis, including the impaired protein degradation, the mitochondrial dysfunction and the oxidative stress, neuron inflammation, and also the impaired intracellular membrane trafficking. As shown in this figure from nice review of errors, we can see a number of PD linker genes are involved in multiple steps of this endosomal trafficking pathway, starting from endocytosis on the plasma membrane to the downstream endosome-lysosome trafficking, and also the autophagy pathway. So in our lab, we are interested in the endocytosis, particularly the endocytosis at the presynaptic terminal to regulate the synaptic vesicle recycling. So in general, after the release of neurotransmitters like dopamine through the exocytosis, the synaptic vesicle components, including both lipid and proteins, need to be very efficiently and quickly recycled to form the new synaptic vesicle pool. And this recycling pathway is mainly regulated by the class 3-mediated endocytosis. And the two very important PD-linked proteins involved in this pathway we are studying in the lab is SNAPGenin1 and DNA-JC6 genes, which encode the protein SNAPGenin1 and oxalin. And these two are recently linked to the autosomal early onset Parkinsonism, and giving the name as PARK19 and PARK20. So here's the cartoon to show you the multiple steps of the class 3-mediated endocytosis at the presynaptic terminus. So the first step actually is not shown here, is the sorting of the cargo and the recruitment of the class ring and its adapter proteins to the endocytic site. And then the plasma membrane will be evaginated to form this called class ring coated pits on the plasma membrane. Next step, the dynamine small GTPs will be recruited to the neck of the pits to trigger the fission of the vesicle from the plasma membrane. And after that, the class ring cage needs to be removed to generate the nascent and naked vesicle, so the neurotransmitter like dopamine will be repackaged or reload to the lumen of the vesicle. So for this class ring uncoating step, there are two important protein complexes involved. The endophyllin SNAPGenin complex is recruited earlier to remove the inner layer of the class ring adapters by depleting the PIP2 on the plasma membrane. And later on, oxalin, which is a cost chaperone, work together with the HVC70 to disassemble the clustering cage, the outer layer, to completely remove the clustering coat. So I will start to talk about snapgenic first, which is a published story during my postdoc. So as I mentioned, this is a lipid phosphatase using the PI45P2 on the plasma membrane as the substrate. It has a SUC1 N-terminal domain and the central 5-phosphate domain. These two lipid phosphatase domain sequentially dephosphorate the PI45P2 and release the clustering adapter like AP2 for uncoating. From Pietro's previous study, we already know the complete loss of function of snapgenic resulted in early death and very severe epilepsy, as shown in our snapgenic knockout mouse model. And recently, in the human patients who carry some homozygous nonsense mutation of snapgenic, they also suffer from seizures and show the early death during childhood, suggesting the snapgenic is a very essential gene for the neuronal function. Interestingly, back to 2013, our collaborators identified the first missense and pointing mutation is R2Q mutation in the SUC1 domain of snapgenic that is linked to Parkinson disease. So the patient carrying this pointing mutation, they show this recessive early onset of Parkinsonism. So only 2 copies of the mutations developed the phenotype, and they developed the Parkinsonism at early, about 20 to 30 years old. And in addition to the typical motor symptom, they also suffer from seizures. So previously, we demonstrated that using the in vitro phosphatase activity assay, this point mutation selectively abolished the SUC1 domain's activity to the substrate PI4P. The central 5-phosphatase activity is intact, suggesting this missense mutation leads to the partial loss of function of snapgenic. To further study the cellular mechanism, we generate the knock-in mice carrying this missense mutation of Parkinson's disease. Very interestingly, these mice recapitulate the human patient manifestations. First is a recessive, so only the homozygous knock-in mice show the phenotype. 20% they would die before weaning after 3 weeks of birth, and they show this abnormal posture. The rest can survive to the adulthood, but they all suffer from these tonic chronic seizures at a young age, about 1 to 2 months old, which is also reported in the human patients carrying the same mutation. We also performed the motor coordination behavior test, like the rod roll and balance spin, and showed that indeed there's impaired motor coordination in this knock-in mouse model. So this provides us a good model, animal model to study the pathogenic mechanism. So we next checked the subcellular localization of this endocytic protein at the pre-synaptic terminus using the cartilage cortical neurons. So the immunostaining show that although the snapgenic protein level is unchanged, its localization at the synapses is significantly affected. You can see this mutant snapgenic abnormally clustered or accumulated at the pre-synaptic terminus, as shown as this puncture along axon compared to the Y-type neurons. We also check several other endocytic proteins and observed very similar clustering cluster phenotype for these proteins, including the oxalin, another uncoating protein I mentioned, and also the clustering and its adapter AP2. We further demonstrate by triple staining with the excitatory and inhibitory pre-synaptic marker, and found that these endocytic clusters are predominantly aggregate in the GABAergic synapses, but very few in the excitatory glutamatergic synapses, suggesting there's the impaired balance between excitatory and inhibitory transmission, at least in the cortex. That probably can explain the epileptic phenotype in both patient and the mouse model. We also performed the electron microscope to check the ultrastructure of the synapses. As you can see here, in the knock-in synapses, actually there are a lot of vesicles that are still coated with this clustering cage, as shown here, compared to some smooth naked snap vesicle in the Y-type. So the quantification clearly shows the clustering coated vesicle number is significantly increased and consequently, the synaptic vascular number is decreased. So together, those morphological evidence strongly suggests that this single missense mutation in the synaptic journey lead to the clustering uncoating defect to slow down the synaptic vascular recycling. In collaboration with Tim Ryan's lab at Cornell, we also performed this pH lowering-based axon and endocytosis assay. As you can see here, under different electrostimulation, the increased this pH-sensitive GAP signal, which represented the exocytosis rate, is unchanged. However, after stimulation, the recovery of the GAP signal, which represented the endocytic rate, is significantly delayed, supporting our morphological evidence that indeed the synaptic vascular recycling is slowed down or delayed. So how about the dopamine neuron? Because this is a PD linked mutation. So we checked the dopamine system. Here is the cartoon to show you in the midbrain, there are two major subtypes of dopamine neuron. The one from subtrunction nigra sends the axon to the dorsal striatum to control the motor function. This so-called nigra striatum pathway is selectively degenerated in PD. However, the VTA dopamine neuron sends the axon to the ventral striatum or the cortex to regulate the reward function, which is not effective. So actually, this is the most challenging question in the field. Why there is a selective nigra striatum degeneration for this subtype of dopamine? So we first check the cell body in the midbrain using the tyrosine hydroxylase as a DA marker. But basically, we don't see very obvious loss of the dopamine neuron in either VTA or nigra, and the morphology of the cell body and the neurite and the dendrites are fine. However, when we focus on the axon terminal of the dopamine neuron in the striatum, we found a very interesting phenotype. So this time we also use another dopamine transporter, the marker for dopamine terminals, to label together with TH. As shown here, we found very interestingly, there are some co-aggregation of these two dopamine markers, very specifically in the knock-in striatum compared to the wild type. And more interestingly, we found only in the dorsal, but not the ventral striatum, we can detect these protein aggregates. So this is the overview. Hope you can appreciate. Most of the dead positive clusters are present in this dorsal lateral striatum, which receives the axon input from the nigra neurons, but the ventral part is fine. So we find this result is very interesting and important because this perfectly mimics the selective degeneration of the nigra striatum pathway. And importantly, this is kind of axonal or synaptic degeneration for this type of dopamine neuron. So providing a very good tool to study why the endocytic dysfunction selectively affect the synaptic trafficking or transmission in these dopamine neurons. And finally, we also collaborate with Eddy's group at Yale and performed a vatimetry experiment to measure the dopamine release in vivo. So when we stimulated the cell body of the dopamine in the midbrain and measured the dopamine release from striatum, we found, again, under the high electrostimulation, the evoked dopamine release is significantly reduced in the knock-in mice, suggesting the dopamine synaptic transmission is impaired in this model. So here is a working model for this part. So we find the first missense mutation, which caused a partial loss of function of synaptic journey and impaired the clustering uncoating for synaptic vascular recycling. Next, I will spend the rest of the time to talk about an unpublished story focusing on another clustering uncoating protein called oxalin. So as I mentioned, oxalin is a co-chaperone highly expressed in the brain. It contains the P10-like domain, which can bind to the lipid on the membrane, the clustering binding domain to recruit clustering, and also the J domain, the C terminal, to recruit the SHC70 for the clustering disassembly and uncoating. Here, at least so far, the PD-linked mutations in oxalin genes throughout the whole gene, and most of them are splicing mutation or truncated mutation, which will lead to either partial or complete loss of function of the oxalin. And if we compare the PARK19 and PARK20 patients, actually they share very similar clinical symptoms. As mentioned before, they both are early onset recessive Parkinsonism. They have motor symptom and epilepsy, which is atypical symptoms. And the brain imaging also show, like the DET scan, show there is a dopamine deficiency in the striatum, suggesting there is a terminal defect. So we decided to use our oxalate knockout mouse model to study the underlying mechanism. So again, interestingly, similar to the synaptic knock-in mouse model which I just showed, these mice also recapitulated the patient manifestations. So only homozygous mice show the neurologic phenotype, including the hind limb clasping, which is a sign of neurodegeneration. They have relatively mild motor symptom at a young age on the balance spring test. And they also suffer from the epileptic seizures as shown in this video. So at the cellular level, when we culture the cortical neuron, we find very similar this endocytic clustering phenotype at the presynaptic terminus in the oxalate knockout neurons. And again, it predominantly cluster in the GABAergic synapses. We explain this potential epileptic seizures there. And more interestingly, if we check again the dopamine system in the striatum, particularly the terminus, we found exactly the same phenotype, like the SNP21 knock-in. You can see both TH and the DET, they co-aggregate very specifically in the dorsal striatum, but not the ventral striatum. So these two independent endocytic mouse models show very selective negral striatum axon degeneration, which is very interesting. We next crossed the oxalate knockout mice with our SNP knock-in mice, and the double homozygous mice actually show very strong genetic interaction. You can see from this survival curve, they have much shorter lifespan compared to the single mutant, suggesting there's a synergistic function between these two endocytic proteins. We also showed, again, in the cultural system, the double mutant, they have more severe endocytic clustering phenotype compared to the single mutants. And by the EM, we show there are more these clustering-coated structures at the presynaptic terminus, including both clustering-coated vesicle and empty cages, suggesting the loss of function of these two genes worsen the endocytic defect, particularly the clustering uncoating defect. Again, in the striatum, we found this dystronic dopamine terminal phenotype. Now we can observe in both dorsal and ventral striatum in the double mutant, but not in the single ones. So just to remind you that this part, the ventral striatum actually receives the axon input from the VTA dopamine neurons, suggesting in a double mutant model, both types of dopamine neurons are affected. And we also try to study the nature of this protein aggregates. And by the EM, including the immunogold labeling, we found this very strange or abnormal multi-layer membrane accumulation in 3 mutant genotypes. So there are multiple layer membrane accumulation next to the cell body of the striatum neurons. And this is a 3D EM to show really there's a massive accumulation of the membranes in this abnormal structure in the mutant striatum. And we are still working on this part to try to figure out what's the structure and how it's triggered. And we have some preliminary evidence suggesting this membrane accumulation actually is connected to the plasma membrane, suggesting there's a massive endocytic defect in the dopamine terminus as well. We also try to examine several other endocytic or synaptic protein to see whether they also co-aggregate in these abnormal structures. One of them we find is the AADC. You can see this one co-aggregate with that perfectly in the mutant striatum. And AADC actually is the second enzyme work together with TH to convert tyrosine to the dopamine. So this suggests probably there's impaired dopamine synthesis in this abnormal terminus. In addition, we also found another interesting synaptic classical protein called the SV2C is also co-aggregate with DAT or TH in all the genotypes. Very interestingly, this glycoprotein on the synaptic classical is enriched in the basal ganglia. And last year, actually, this one was identified as a novel PD risk gene by GWAS study, suggesting probably the membrane trafficking of this SV2C is also impaired in our mouse model. And finally, we also observed some interesting adaptive change in the mutant striatum, in addition to the abnormal clustering phenotype in the dopamine terminus. As shown here, in addition to see this TH and that double positive clusters, which represented the dopamine terminus in the striatum, we also observed some TH positive cell bodies, which is negative to DAT or negative for DAP32. So they are not the midi spine neuron. Actually, people think this is a TH positive interneuron in the striatum. And we also clearly demonstrate another enzyme, AADC, is co-expressed in these interneurons. And you can see the number of the TH positive neuron is significantly increased in the double mutant. So if you search the literature, actually, this C neuron has been reported earlier in some neurotoxin-induced PD models. For example, the TP-treated monkey or rodent, you see the significantly increased C neurons. So the idea is that probably there's a local dopamine synthesis try to compensate this dopamine deficiency due to the under the Parkinson disease condition. So we're still testing the hypothesis in our model. We also find another adaptive change. So as again, hope you can still follow. So this SV2C and TH positive, the yellow clusters is the dopamine terminal aggregation. So we found the TH positive actually is a GABAergic interneurons. In addition, we find the third type of another type of interneuron which highly express SV2C in their cell body. Actually, this neuron, when we use a chat to label them, actually this is a cholinergic interneuron in the striatum. And this is a quantification to show you in the double mutant, the striatum, the SV2C, which is a new PD risk gene, the protein level is significantly increased in this cholinergic neuron as well. So this is also a very interesting observation because the cholinergic neuron, the interneuron in the striatum, actually play an important role for this microcircuit in the striatum. There's a crosstalk between the acetylcholine and dopamine signal, and the balance actually is impaired in PD with the hyperactivity of the acetylcholine signal. So now one of the projects ongoing in the lab is to study how and what's the function of the SV2C in these two presynaptic terminals to regulate their crosstalk. So in summary, we developed and characterized two independent endocytic mutant mouse models, which phenocopy each other and recapitulated the human patient's manifestations. At the cellular level, they showed the clustering uncoating defect to slow down synaptic reticle recycling. And more importantly, they show very selective axon degeneration in the negral dopamine neurons. And the double mutant show the additive effect and the synergistic function. They have shorter lifespan, more severe endocytic defect, spread this striatum terminal defect, and also the adaptive change in the interneuron and the striatum. So in the lab, in addition to work on these two mouse models, we'll also study several other PD-linked proteins function in this pathway. For example, how Parkinson's ubiquitination regulated the vesicle recycling, or lactose phosphorylation to regulate the different endocytic proteins function. So the key question we try to address is why this generic synaptic defect selectively degenerated the dopamine neurons. So one of the hypotheses we're trying to test is this cytosolic dopamine accumulation. So the Dimitri Krank's lab did a very nice work to show the accumulation of the dopamine in the cytosol causes the oxidized dopamine, which is toxic, and they induce the downstream mitochondrial or lysosomal dysfunction and alpha-synuclein accumulation. So actually, the impaired synaptic vesicle endocytosis can explain very well this hypothesis because uncoating defect or lack of the reformation of the synaptic vesicle will cause the accumulation of the cytosolic dopamine. So that's the hypothesis we're currently testing in the lab. So I would like to thank my lab for all the lab members' hard work, particularly a talented research assistant, Mian, and she did most of the work for the unpublished story with help of Yonan and Sidra. And I'd also like to thank great collaborators from Singapore and overseas, including my postdoc mentor Pietro, and also the funding source to support our research. So my lab is still young and energetic. So if anybody is interested in studying cellular mechanism of neurodegeneration, please consider joining us. Okay, I'll end here and I'd like to take some questions.
Great, thanks, Mian, for a great talk. Interesting. Now, other questions? I might have a quick one. So I'm trying to learn. So it's very interesting, the pre-synaptic trafficking the defect. But how do you expect to how this presynaptic failure may be related to the death of dopaminergic neuron? Do you have any clue yet?
Yeah, that's a good point. Yeah, in our mouse model, at least the single knocking, we check in the midbrain, and there's no degeneration or loss of the cell body. But we think this is kind of the synaptic degeneration is an early event in the PD.
Sure.
I think maybe combined with the neuron toxin model, maybe we will see some loss of the cell body in the midbrain. So that will
great. Sure. Question from Sheng. Please.
Hi. It's a great talk. It's very interesting. Sorry for it.
There's some echo here. Yes. Okay.
Sorry for it. So it's very interesting. So your result basically two questions. So one is your results suggest that this is auxin, and then this other gene. When you have these two double mutant together, you have more severe phenotype, right? That means these two genes actually function in the likely parallel pathway because your codes are not on. Is that right? So that they both working on the decoding. Is that the first on encoding? Is that true?
Yeah, that's a good point. I mean, the geneticists will think about if they have the additive effect, that means they probably work in the parallel pathway. But like in the fly model, the Parkinson and PINK1 double mutant, they have similar defect as the single one. But here, I think they work on different steps of the clustering encoding. Synaptic journey plays a role for the lipid removal and the adapter release. And the oxalin actually is for the next step, the clustering cage disassembly. So yeah, we clearly show the genetic interaction there, and also the cellular mechanism is clear. They work on the same pathway. So I think it's still not in parallel. It's just a different step or different stage of this endocytosis.
Okay, so the second question is about this TH accumulation. I assume TH, this is enzyme normally accumulated in the cell body, right, in the cytosol, but you show that they get accumulated in the exon terminal. That's kind of like stacking. So that looks like this protein get either like trafficked to the wrong place or get a. So what's your explanation of that?
Yeah, let me show you again. Yeah, it's very interesting. We found several dopamine-related proteins or enzymes accumulate in these clusters, including the DAT and the cytosolic enzyme TH, and this vascular protein SV2C, and SNAP25, another Snell complex. We think it's because there's a massive membrane accumulation, actually probably evaginated from plasma membrane, and this wrapped everything at the presynaptic terminal, including the membrane protein and the cytosolic enzymes. So that's why you can see the TH that they co-aggregate at the nerve terminals.
So that means they are not that specific, right? This kind of abnormal accumulation is not very specific. It's just
probably any protein enriched at the presynaptic terminal, you will detect it in these abnormal structures.
Okay.
We're still testing this. And we're also testing whether in these clusters, the TH and AADC, they are still active, like whether they can still synthesize dopamine or it's a loss of function there.
Okay. So do you have any other marker that show that you have more extra, for example, plasma membrane biogenesis? That's why you have such excessive membrane accumulation. Do you have any insight on it?
At least the SV2C, we think is a synaptic vascular protein. But after exocytosis, maybe the impaired recycling, this protein will accumulate on the plasma membrane. But that is a very it's a well-known plus membrane marker on the terminal of the dopamine neurons.
Okay, it's very interesting. Thank you.
Yeah, thank you.
Great. Tao Hui, please.
Hi, it's very nice talk. I'm curious when this presynaptic trafficking is affected, and what happens to its corresponding post-synapse? Because when this does this clustered protein also affect the postsynaptic part? Have you examined it?
Yeah, actually, we only checked the DAP32, which is a marker for the striatum midi-spine neuron, and we don't see abnormal morphology of the postsynaptic neurons. But yeah, definitely we need to do more like a physiological recording. Actually, we also checked the dopamine receptor, I think, the D2 receptor. We don't see any defect there.
Okay,
but the whole striatum is messed up. As I showed here, in addition to the cluster in the dopamine terminals, there are also adaptive change for the GABAergic interneuron and the cholinergic interneuron in the striatum. And we also have some evidence that the glial cell is also messed up. So we are still doing some mass spec-based or the single cell sequencing to try to figure out. So we don't know. We think the primary defect is from dopamine neuron. And those kind of striatum change is the secondary or cell anatomous effect. So we're still working on this to figure it out. Yeah.
Okay, all right. Thank you.
Yeah, thanks.
Okay, one more question from Sheng, please.
Thanks. So if no one asks a question, so a follow-up question. You show very striking this dorsal ventral difference in terms of dopamine neuron, this pathology. Do you know what's the reason behind that? They are all dopamine neuron, right? But then like why the ventral have no effect, but dorsal have such a striking effect?
Yeah, exactly. This is the major question we try to address in the lab. So we have two single mutant mouse model shows very selective dorsal defect. But now the double mutant, you see actually this phenotype spread to the ventral, even the bottom of the structure. So we think the A9 dopamine neuron in the nigra or A10 dopamine neuron in the VTA, they are both sensitive to the endosity defect, but they're at different threshold. So in a single mutant, maybe somehow the A9 nigra neuron, they are more sensitive. So they start to show this phenotype. So if you have the double mutation, which has the additive effect, both subtypes are affected. I think at the end of the talk, I mentioned some hypotheses like the dopamine accumulation in the cytosol. Probably that's the feature from nigra neurons. So that's why maybe there's accumulated cytosolic oxidized dopamine to induce a neuron toxicity in the dorsal striatum specifically.
Okay.
People have been doing this kind of single cell sequencing to check gene expression difference. Have you checked and see whether there's any major difference between these two clusters?
This is next step. Yeah, actually, we're collaborating with another group at the Duke to use the single cell technique to figure out in both midbrain and the striatum to see the dopamine neuron subtypes difference and also different striatum interneurons gene expression pattern.
Okay, thanks. Great talk.
Okay, thanks, guys. So we have to move to Lee's talk. Hi, Mian. There's one more question, related question, I believe, in the chat bar.
Yeah, maybe
you might type in the answers. Yeah.
So I think Suboj, we need to have Subojit's question.
Sure, great. Go ahead. Yes.
Right. Yeah, it's a great talk. I really enjoyed it. So sorry for my video. But my question was that these aggregates that you're seeing after the mutations, I was wondering what exactly those aggregates are. Are they really protein aggregates of like all the other synaptic proteins, or are these some sort of vesicle aggregates that have molecules around them? I mean, I know from your EM, it doesn't sound like these are vesicle aggregates, but
yeah, it seems very interestingly. So we still don't know. That's why we are trying to do some claim, the correlative light and the electron microscope. The immunogold already show that TH is accumulated there. So
these are in the synapses. Oh, okay, I didn't get that. You see, these, these remind me of these onion skin myelin sheets, but obviously it can't be myelin because it's inside.
We don't think it's myelin sheet or autophagosome because I think
autophagosome.
But yeah, I didn't show the preliminary 3D reconstruction. I think this membrane accumulation, the onion-like one, actually connected to the plasma membrane. We think somehow there's no clustering-coated vesicle accumulation there in the dopamine terminal. We standard clustering and adapters, they don't coagulate there. So we think there's probably another type of maybe bulk endocytosis defect to cause this massive membrane evagination.
I see. And then it would make sense that this very complicated membrane is just recruiting the other proteins non-specifically and creating that accumulation. Okay, I got it. Thank you.
Actually, in the literature, one of the oxalin transgenic mouse model, they also by the EM show this similar membrane evagination.
Yes. Thank you.
Okay, thank you.
Great. I learned a lot. Sorry. Yeah, thanks, man. You learn a lot. Now, okay, now we're ready for move on to this talk.
Okay.
Aaron,
thanks,
everybody.
Thanks. Yeah, great talk. Okay, it's my great pleasure to introduce the next speaker, Li. Li is actually my across the street neighbor, and I never, here in Shanghai, I never knew it until I was just out walking my dog, and I ran into one neighbor, and she said, "Oh, there's a world famous neuroscientist living around here." And I said, okay. And then I said, who's that? And she said, her name's Li, and she's a professor at NYU Shanghai. And so I quickly looked her up, and then she's in psychology. And my best friend is in Beijing, who's a professor at Beijing Normal University. So I messaged my friend. I said, do you know this person? She said, oh, I'm actually going to meet with her tonight. I said, oh, maybe you can connect us. And then she connected us on WeChat. It's a Chinese messaging app. And then Li said, oh, why don't I just come across the street and I can buy you some bubble tea. I said, I'm not eating sugar anymore. But we had coffee, and then we just got to know each other. And then Shanghai was locked down, and it's just been great to have a great neighbor and friend. I have an apartment here, but she has like a very fancy luxury townhouse. I think it's like right across there. You can see it. And so we've been having barbecues and fun things during this lockdown. So there's a silver lining there. Okay, so we'll scour the earth for the best NeuroZoom speakers. Sometimes they're literally right across the street. So Li did her undergraduate degree at an elite Chinese university, the #2 university in all of China, Peking University in Beijing. Then she came to the US to do a master's degree at Stony Brook, and then did a PhD at Brown University in cognitive science. She did a postdoctoral fellowship at Harvard Medical School, and then she even worked at NASA, the NASA Ames Research Center, which is in Mountain View, California. And you'll sort of see her expertise and focuses on visual perception. She then moved to the University of Hong Kong, where she rose to become an associate professor. And now she's a professor of neuroscience and psychology at NYU Shanghai. And she's editor of many journals in her field. Her major research focuses on studying how humans perceive information in the world, and how that perceiving that information is used to control movement. She uses a wide range of experimental approaches, from virtual reality to brain imaging, and she studies hand-eye coordination. This is the normal situation. She also uses patients who have brain damage or who have diseases and how that can affect visual. She studies elite baseball players to see how hand-eye coordination can contribute to improved batting. So really the wide range of topics. So looking forward to hearing the latest. Li, take it away.
Hi. Thank you, Aaron, for the nice introduction. First, I want to say I'm really not world famous. And thank you all for coming to my talk. So I'm going to move away from the molecular level to the behavioral level. So the topic of my talk today is impaired ocular tracking and cortical atrophy in idiopathic rapid eye movement sleep behavior disorder. It's a really long title. And I want to mention that my lab, we study visual motor control, the perception and control of self-motion and related neuromechanisms. And so this study is based on a previous study that examined the eye tracking, the ocular tracking abilities in athletes, specifically baseball players. And we found that the ocular tracking abilities are pretty much, it's an untrained potential that actually can predict their future performance. So now we're applying this paradigm to study the patients with neurodegenerative diseases, especially with motor dysfunctions. And this study is a first step, and the data already published, Journal of Movement Disorders early this year. So yeah, this actually is going to be a short talk, not as many rich data as the previous talk, but I hope that you enjoy it. Okay, so before I start my talk, I want to show you a picture of Shanghai. Since I'm from NYU Shanghai, you might be wondering where it's located. So this picture shows the 20 years growth of Shanghai, pretty splendid, from 1987 to 2013. And NYU Shanghai is located in the central business district, the downtown area. And Shanghai is under lockdown for over 2 months, almost 2 months. And this is the current picture of the central business district, pretty much nobody. Okay, for your information, for your interest. Okay, so I'm gonna move on to the talk. So first, a little bit background information. So what is a rapid eye movement sleep behavior disorder? In short terms, it's a RBD. It is involves, I want to describe the symptom first, involves abnormal behavior caused by a loss of physiological motor inhibition, usually present during the REM sleep. So you know the sleep have different stages, and there's one important stage that is related to our rejuvenation after sleep. It's the rapid eye movement. So I'm going to play a video to show the major symptoms of RBD patients. So from the video, you can see that the violent behavior can be quite dangerous because the patient can hurt him or herself and can also hurt bad partners. Okay, I want to mention that the idiopathic RBD, so the RBD without specific origin, it pretty much affects 2% of older adults. So it's very prevalent. And it's not just a sleep disorder. It's an early signs of several neurological diseases, including Parkinson's disease. And it's actually the Lewy body dementia and multiple system atrophy. Okay, so the PD, kind of the development from RBD to PD is actually most frequent. And here is a graph that shows the neurological disease free survival rate against the time from RBD diagnosis. So you can see that pretty much after 5 to 10 years of diagnosis of RBD, about 80% of the patients would develop a certain neurological disorder. Okay, and at about 15 years, pretty much over 90% of the patients would develop a certain type of neurological disease. So the conversion from idiopathic RBD, IRBD, to neurological diseases is a remarkably high risk, and there's a notably long interval. Okay. And also I want to mention that the sign of RBD is really kind of, so PD, the most important symptom is motor symptoms, as mentioned in the previous talk, right? But the IRBD can be shown like decades before the motor symptoms in PD patients like showing up. Okay. So I want to mention the motivation of the current studies. So given the long disease progression, it's important for us to find early alterations in behavior. For example, eye movements, and related to the brain structural changes, because so it's important to know like the conversion of the IRBD to neurological diseases so that we can develop early intervention and to delay and even kind of, prevent the kind of the conversion from IRBD to neurological diseases. So why do we study eye movements? I mean, first, it's highly sensitive and it can be objectively measured, unlike mostly use the clinical questionnaires. So ask the patients to self-report their symptoms and can be very subjective. And eye movements can be complex enough because eye movements involves many neural networks in the brain for studying both the cortical as well as the subcortical regions. And it's simple enough for patients to perform because the patients usually they have pretty slow cognitively. There just can be certain problems, and then you ask them to perform a complicated task. It's usually very difficult, but eye movements is a very simple task, especially the task we're using. Very simple to perform, and it's only 15 minutes, and it measures many aspects of ocular tracking abilities. Okay, so the purpose of the current study is to examine whether the ocular tracking ability related to eye movement is impaired in early stage IRBD, and we use a 15-minute ocular tracking task that can provide, again, 12 different oculometric measurements. Okay, and then we're going to examine the gray matter alterations associated with the ocular tracking abilities in IRBD, and we pretty much use the T1 weighted scans of brain imaging. And the last, most importantly, we want to examine the predictive power of our measurements, the eye tracking, the oculometric measures for conversion to neurodegenerative diseases in IRBD. And the three well-known clinical biomarkers for early diagnosis include olfactory loss, non-motor symptoms, and autonomic symptoms. So the first one is objective, can be objectively measured, and is of the highest sensitivity. And the latter two, the biomarkers, the non-motor symptoms and the autonomic symptoms, are pretty much reported from the self-reported questionnaire, so they're quite subjective and not as sensitive as the olfactory loss. Okay, so first I want to describe the patients, participants in the current study. We have a relatively large cohort of our IRBD patients as well as age-matched healthy controls. And a subgroup of our IRBD patients, they also participated in the brain imaging study so that we can evaluate the related to brain structural change with their ocular tracking, like impairments in ocular tracking abilities. And then this table lists the characteristics of our participants, the patients. Okay, so the disease duration, the disease from onset, the disease duration from diagnosis, and these two are the UPDRS is a common clinical scale used to evaluate Parkinson's disease patients. This one, it's a cognitive cognitive questionnaire to make sure our patients are not cognitively kind of deficient. Okay. So next, I want to describe the ocular tracking task that we used. Again, so this is a very simple task, but can provide a lot of data that reflect the different aspects of our ocular tracking abilities. So this is a setup. It's actually setup involves eye tracker, sophisticated eye tracker, and monitor. And so the participants, they put their head on the chin rest, and then they watch a movement of a red target. So at the beginning of the trial, the red target stays in the center of the screen, and then it steps back for a short amount of time, and then moves at a constant velocity in one direction. And then, so the kind of direction we tested ranges from 0 to 360 degrees, and the velocity ranges from 16 to 24 degrees per second. Okay, and this is based on a previous established paradigm, but we perfected it, kind of make it more make it more efficient, as well as suits the kind of data analysis to make sure the data are reliable. Okay. So here I'm going to use a video to illustrate on each trial what the patients, the participant is supposed to do. You look at red dot. and then it's going to move, and you just use your eyes to track the movement of the dot. Very simple. I'm going to show you another trial. Very simple task, right? And then I want to say that for the structural brain imaging analysis, we performed morphometric analysis, surface-based, that includes the cortical thickness analysis, surface area analysis. For the subcortical analysis, we pretty much evaluated the volume. Okay. So I'm not going to bore you with the details of this table, and the fonts are too small for you to see it clearly anyway. It just describes the procedure that we used for this study, kind of the statistical analysis. So we did all the clinical assessments, make sure they have this disease, of course, and make sure it is early stage, and make sure they don't have any other kind of brain diseases or cognitive disorders. And then subgroup of the IRBD patients, they participated in the structural MRI scanning, brain imaging, and later on we did, analysis, like correlation, and to find the relationship between the behavioral measurements and the brain imaging data. Okay, so now I want to show you a data from a sample data from a typical healthy control subject. Okay, so I want to draw your attention to the graph here. There are 12 measurements. Okay, the indices, they measure 4 independent aspects: the pursuit initiation, steady state tracking, direction tuning, and speed tuning. So regarding the tracking process, there is pursuit, the initiation, right? Start the eye movement, the process. Steady state tracking. So your eyes, the target moves, and your eye is going to track the movement of target. So there's initiation of the eye movement, okay. Pursuit eye movement. And then the target's moving at a constant velocity. Your eye is smoothly tracking the target. We call it steady state tracking. Okay. So regarding the target movement, whereas direction tuning is how accurately your eye movement, eye tracking can follow the direction of the target movement. And your speed tuning is how accurately that your eye movement, the speed of your eye movement matches the speed of the target movement. So the 4 relatively independent aspects. So next, I'm going to explain the measurement indices we used in each to measure each of the 4 aspects in more details. So here I'm plotting is the recorded velocity of the eye movement, okay, as a function of the time, okay. And so you can see the motion onset is when the target starts to move, and the pursuit onset is where the eye starts to move, because targets move, we have a reaction time, right? And then the target moves, the eye starts moving, and then there is acceleration, because eye first is speed is 0, and it needs to accelerate to a constant speed, and so it can smoothly track the movement of the target, right? So this is the acceleration stage. You can see this part is also accelerating, and then the last part indicates steady state. The target is moving at a constant speed, and our eyes are also moving. The match cannot be 100%. So our eyes is smoothly tracking the movement of the target. But then at a certain point, it lags behind the movement of target, and then it has generated saccades. So there are 2 types of eye movement: smooth pursuit. I can pursue the target very accurately. I do saccade, this jump of the eye movements indicated by the peak, the red lines, the red peaks here. Okay, so we call it catch-up saccade. So that I can jump, and then I can follow the target again, and then I can't follow the target anymore. I jump, okay, generate another saccade. Okay. So this is the steady state. And so for the pursuit initiation, we have 2 indices. The latency measures the time between the target movement onset and the initiation of pursuit. And there's acceleration, which is the mean radial acceleration in the first 100 milliseconds interval following the pursuit onset. And in the steady state tracking, we have gain, right? Steady state gain, which is mean pursued eye speed projected along the target movement direction and divided by the target speed. Okay. And we have proportion smooth, which means during this steady state tracking, what percentage of the time is corresponds to smooth pursuit tracking. Okay. The larger the percentage means the person's smooth pursuit abilities is better. Okay. And the latency is that shorter is better, acceleration the higher is better. Okay. And then so in the steady state tracking, there is direction tuning. So here I'm plotting the pursuit direction as a function of the target direction. And then there is direction noise, which indicates the precision of eye response to direction. And there is direction anisotropy, which indicates the differences in the ability to discriminate between different directions. So there's up-down asymmetry, whether the upward eye movements and the downward eye movements, where there is asymmetry, and then there is oblique effect anisotropy, which means whether your tracking ability along the oblique directions is better or worse than your tracking abilities performance along the cardinal axis. Okay. And then there is speed tuning. So here is the pursuit of eye movement speed as a function of target speed. So there's speed noise that measures the precision of eye response to speed. And there is speed responsiveness, which is the best fitting linear regression slope of the mean radial pursuit speed, which is the slope of this regression line. And it indicates the ability to discriminate between small differences in speed. Okay, so these are the measurements that we took to holistically examine a person's ocular tracking abilities. So again, it's a 15-minute task. It's very simple, but it can provide us with a lot of data that we can do. We can do a lot of analysis on. Okay. So here, what I'm showing is sample data from a typical healthy control and a typical IRBD patient. So I want to draw your attention to the pursuit initiation. So you can see the latency shows a lot of difference here. Okay. And so what I'm plotting here is this red bar. The red line here indicates the median, and here is the distribution over 180 trials, and this is the latency. You can see the whole distribution for the IRBD patient. So this is data from one subject, okay, and then it's shifted, which means the IRBD patient has longer pursuit initiation. Okay, I'm 20 minutes. I'll try to speed up. And acceleration, the same thing. It's actually IRBD has smaller for this specific patient, smaller values. And then also I want to draw your attention to the steady state tracking. So you can see the gain for the normal, for the healthy control is higher than the patient. And the proportion smooth is actually similar. And saccade amplitude, the catch-up saccade amplitude, the patient is higher, which makes sense, right? Because they can't accurately pursue the moving targets, so they have to generate saccade, and amplitude is larger. The direction tuning here, there's some difference, but statistically it's not significant, and there's not much difference in speed tuning. Okay, so I'm going to move on to the next slide. So here I'm plotting the difference between the healthy controls and IRBD patients for the 4 measurements that showed statistically significant difference. There are 2 from pursuit initiation and 2 from steady state tracking. So in summary, the ocular tracking ability in IRBD patients is inferior to that in healthy controls in both the pursuit initiation indicated by the longer pursuit latency and lower open loop acceleration. And also in steady state tracking indicated by smaller steady state gain and larger amplitude in catch-up saccades. Okay, so these are the behavioral data that clearly shows a difference between the IRBD patients and the healthy controls. So next, this one, I want to show you the results from the structural brain imaging. Okay, so this one, the top part shows the right hemisphere of IRBD patients. The highlighted area are visual area B4 and LO3, the lateral occipital 3. Okay, so these two areas, so the IRBD patients displayed cortical thinning in these two areas. And also for the left hemisphere, this area showed surface expansion and compared with healthy controls, which means that, you know, these three areas compared with healthy controls, they showed significant difference. Okay. And then out of these three, the cortical structure changes, only this one, the right hemisphere V4 is correlated, the changes is correlated with the behavioral measurements in terms of the, for both pursuit latency as well as the open loop acceleration. Okay, so left graph plots the pursuit latency as a function of cortical thickness. You can see that the red dots indicates the data from from RBD patients, and the blue dots are data from healthy controls. So you can see there's significant trend for the RBD patients, but this trend disappears for healthy controls. And the same thing for the acceleration data. So this indicates there's, the red dots indicates data from RBD patients. There's significant increase. So which means the larger the particle thickness, the higher the acceleration, but this trend is absent in healthy controls. So in summary, the cortical thinning of right V4 in IRBD patients was correlated with deficits in the initiation of pursuit eye movements in terms of both the pronounced pursuit latency as well as a decreased open loop acceleration. So this one, then we explored the relationship between the impaired oculometric measures and clinical biomarkers. Specifically, we found correlation between the olfactory loss, which is the objective and most sensitive biomarker for the conversion of IRBD to neurological diseases such as PD. We found that so the increased olfactory loss is associated with prolonged pursuit latency in ocular tracking in IRBD patients. For the other two, the kind of self-reported biomarkers, we actually did not find any correlation. So what does this mean? I want to summarize it. Okay, so we found that ocular tracking abnormalities, such as pursuit initiation and steady state tracking impairments in patients with IRBD, even early in their disease progression, that have not been reported before. And these abnormalities are coupled with atrophy in brain areas involved in perception of object motion. This is what the right hemisphere, the V4, is known for. And so what this means that the both may serve as additional objective biomarkers for early diagnosis of conversion of IRBD to neurodegenerative diseases such as PD. Okay. And our future direction, I want to mention, as I mentioned, this is the first step of a big project. And we're planning to examine whether the observed the ocular tracking abnormalities and related brain atrophy can show the different patterns for different neurological diseases such as PD, dementia with Lewy bodies, and MSA. And also the next one, I think it's really more interesting, is we want to examine the link between ocular tracking abnormalities in patients with neurological diseases and their genetic and epigenetic variations using a deep learning system. And the goal here is really to provide some genetic biomarkers that are of interest to, the big audience here, the genetic and molecular biologists for the understanding of the neuromechanisms of these diseases. Okay. 28 minutes. I want to say this study is led by my postdoc in the lab, Jing Chen, and we have several doctor collaborators at Reading Hospital. They provided like kind of assessment as well as the data of the brain imaging data. And I want to also thank the funding agencies that support this study. And last, I want to thank you for listening to this talk.
Thanks, Lee. Great talk. we are open for questions. Oh, Hong from Singapore.
Yeah. Can I ask a question about
Please.
so the brain activity affected by IRBD? So you found it's in V4. How about the frontal eye field? Did you find anything?
There's no difference between the patients and healthy controls. We only found 3 areas.
Oh, that's quite surprising because I thought the frontal eye field would be affected because that's directly related to eye movement. So how about other behavioral measurements? Let's say because because rapid eye movement usually related to dreaming, and then for IRBD, did you ask them if they experience a different pattern of dream like nightmare?
This sleep disorder, so their main symptoms is very violent kind of behavior. It's pretty much lack of control of the muscles during REM.
Yeah.
So whether they have different kind of dreams, no, we didn't ask. I mean, so yeah, we didn't ask whether. But why are they relevant here?
Because the other thing is the task you engage them, it's a real, like it's there in normal state, not in the sleep state, right? So have you thought of like ask them some measurement at the sleep, so then you can measure them during their sleep? Heart rate, skin conductance, something like biometric measurement while they're in sleep.
We are mainly interested in visual motor control. So, and the easiest visual motor control behavior we can measure is eye movements, and that's what we did.
Oh, I see. Because it seems very complicated. It's besides of the movement. Go ahead,
so my lab, is specialized in visual motor control and developing this kind of paradigms and data analysis and apply them to, study like kind of the. I want to mention that for the human population, not the molecular level, the kind of neurological diseases, the diagnosis is pretty much based on the doctors, the clinical evaluation. And then when the doctors are able to diagnose those kind of neurodegenerative diseases, it's already late stage. It's not like there is no like objective, very sensitive measurements. And the goal of the study is eye movement is very sensitive. We want to see at a very, very early stage where there is observed differences. There are definitely previous studies that noticed some abnormalities in eye movements in IRBD patients, mainly saccades, but so far there's no single study that has systematically evaluated the kind of eye tracking, ocular tracking abnormalities in such patients. So we actually not only studying the IRBD patients, we also studying Parkinson's disease patients, and then so these kind of behavior measurements. And then the Parkinson's disease patients, their ocular tracking abilities, this project is still ongoing. And later on, we want to see the difference between the two populations. And the lab is also studying, testing actually thousands of kids, their ocular tracking abilities, compared with adults to see the developmental changes in their kind of eye tracking abilities, as well as related to the brain maturation.
I see. So then besides, that's good. So is there any accuracy, fixation, or scan pass difference in their eye movement?
What do you mean? I'm going to go back to.
Are they more accurate, like in their judgment, or their eye movement pattern will be more of error?
So so we have 12 measurements here. Okay. The difference we found is pursuit initiation and steady state tracking. For the direction tuning, which is pretty much measures the accuracy of eye tracking. For direction and speed, we didn't find any difference, significant difference for the sample of patients and age-matched healthy controls that we have in the current study. We didn't find it.
I see. So just in the speed, but not in the direction, you mean?
just in the pursuit initiation, steady state tracking, in terms of the latency, acceleration, steady state gain, as well as saccade amplitude, nothing else, just these 4 out of 12 measures, oculometric measurements we took.
I see. Yeah.
Hey, I have one.
Yeah, thanks.
Hong, are you
Sure,
okay? Hong?
good question.
Yeah, thanks.
Yes.
One question. So you showed that ocular tracking can sort of predict the IRBD, and that seems to correlate with leading to Parkinson's disease. I'm just wondering, in other diseases like ataxia, spinocerebellar, like ataxias, where there are saccades, there are real eye movement defects. Do those patients have sleep disturbances? I'm just wondering, like, if one causes the other, or if this one is just like a marker of things to come for Parkinson's patients. So patients with ataxia that have eye tracking defects as a symptom of their disease, do they have sleep disorders?
That's a very good question. Only 50% of the Parkinson's disease patients, they have sleep disorder. So, I mean, so the IRBD, being argued, is like a biomarker for PD. It's very sensitive, but it's not specific. So that's why, you know, we need to know the person who has IRBD does not mean the person is going to develop PD. But there's, I think, probably at certain age, like over 70, there's people with IRBD, they have 130 times higher risk than people who don't have to develop PD.
I see.
So IRBD, you have to combine with other biomarkers to predict whether the person is going to develop PD.
Got it.
And then these are what I'm listing here are the 3 kind of neurodegenerative diseases that IRBD patients can develop. And PD, I think Lewy body dementia are most frequent. And then these 2, they also show different kind of sensitivity to the olfactory loss. So we measure their olfactory function, right? The PD patients show a loss of olfactory function loss, but Lewy body dementia patients, they don't. So actually the olfactory function, that measurements, they use it combined with kind of RBD. I mean, they use that to sort of predict whether the person, how likely the person is going to develop PD.
Got it. Okay. Thanks so much, Lee, for that great talk. Thanks, Mian, also for the talk, and thanks for everyone for sticking with us.
Yeah,
see everyone next week. Great questions.
Thank you very much for the invitation. Thank you.
Yeah.
Great, thanks to you, Mian. Great, thanks for See you all next week. Bye.
Bye, everyone.
Yeah. Bye-bye.
Bye.